Very good morning and welcome everybody to my talk. At present, I work at the National Primate Research Center of Thailand, Chulalongkorn University, Thailand. My talk today is entitled Genetics Diversity of the two most commonly used non-human primate species in preclinical research and an association with disease infection. My talk today will focus on two species of non-human primates, that is Macaca mulatta or rhesus macaque and Macaca fascicularis or long-tailed macaque, and their genetic diversity, which is associated to the use of them for drug and vaccine development for infectious diseases. These two species of non-human primates are the most commonly used non-human primate species as animal models for vaccine and drug development, especially for COVID-19 vaccine. On the left-hand side of my slide, you will see the process of drug and vaccine development for the step of preclinical trial, in vitro and in vivo, comprising rodents and non-human primates. After the vaccine is confirmed that it is safe and can induce immune response in those animals, the result will be submitted to FDA for approval to proceed to the clinical trials in humans. In this COVID-19 vaccine development process, laboratory animals are very important for toxicity, safety, immunogenicity, and efficacy test. So far, not so many non-human primate research centers in Asia. As a result of increasing awareness of the importance of non-human primates for human vaccine testing, Chulalongkorn University established the National Primate Research Center of Thailand in the year 2012. At present, we have 4 breeding buildings, 525 Thai long-tailed macaques, 1 research building at Animal Biosafety Level 1 and 2, and 1 ABSL-3 module for COVID-19 vaccine efficacy test. Because COVID-19 is airborne disease, which needs the ABSL-3 facility. Our primate center is accredited with AAA International on February 26, 2020, and we were pre-inspected for OECD GLP certified on October 15, 2020. Since May 2020, we have conducted safety and immunogenicity tests for 3 types of COVID-19 vaccine, that is mRNA, DNA, plant-based protein subunit. All these 3 vaccines are safe and can remarkably induce both cellular and humoral immune responses, including neutralizing antibody. In our primate center, we conducted the safety test in compliance with the OECD GLP principle, following the guideline of ICGSK, ICH-M3R2, and WHO Technical Series number 927. Theoretically, the experiments in monkeys were done after the toxicity and immunogenicity test in rodents showed the positive results. After the safety test in monkeys, the sponsors plan to move to the clinical trials in humans for safety and immunogenicity test, and at the same time, the efficacy test in monkeys can plan to conduct in parallel. Why monkeys are important animal models for COVID-19 vaccine testing? On your right-hand side on my slide, you will see the structure of SARS-CoV-2 that causes COVID-19 disease in pink color, and the ACE2 receptors at the human lung in yellow color. SARS-CoV-2 virus binds with ACE2 receptor by using a spike or S protein, the RBD region. At the binding between SARS-CoV-2 spike protein and the ACE2 receptor at the human lung, if we zoom in at the binding regions, 5 amino acids at ACE2 receptor play a major role on binding affinity. From the publication of Luan et al. 2020, they researched in 42 mammal species for these 5 amino acids and binding affinity, and found that ACE2 of mice and rats cannot bind with the S protein of SARS-CoV-2, but the monkeys can. This explains why monkeys are needed for efficacy test of COVID-19 vaccine. Again, by the study of Damas et al. On July 31, 2020, they looked at 25 ACE2 amino acid residues that binds with spike protein of 410 species of vertebrates, from primates to amphibians. They found that 18 species of old world monkeys and great apes have all 25 ACE2 amino acid residues identical to that of humans. They are identified as very high risk for SARS-CoV-2 infection and become the most suitable animal models for COVID vaccine research on efficacy test. So coming back to the 2 species of non-human primates that are commonly used for COVID vaccine testing and are the main focus of my talk today. In Thailand, we have these 2 species of macaques in our country. In general, long-tailed macaque distribute from 20 degrees north to 10 degrees south, and rhesus macaques distribute from 40 to 20 degrees north. If we move to and overlay these 2 maps together, we will see that these 2 macaque species live in overlap at 15 to 20 degrees north, so-called the hybrid zone. In the past 2 decades, there have been many publications reported the hybridizations between these 2 species of macaques by using various genetic markers. In the year 2017, we reported the genetic admixture of rhesus or on this slide ancestry integrates into long-tailed macaque or MF populations using autosomal SNP markers. We found that the hybridization was beyond the hybrid zone, and long-tailed macaques in the north carry higher levels of rhesus genetic admixture than the southern populations. For example, on this map, if we see 2 populations of long-tailed macaque, one is at 16 degrees north and another is at 7 degrees north, it was found that the northern population of long-tailed macaques carry 50% of rhesus genetics, while southern population carry only 15%. By then, we are interested in the susceptibility on infectious diseases of MF that carry different levels of genetic, especially on host and parasite genetic interaction, and co-evolution, and mechanism of action and related genes, and the development of animal models for drug and vaccine testing. So our focus today is tuberculosis. There are reports saying that rhesus macaques are susceptible to tuberculosis when they are infected. 100% of them show the symptom. But In case of long-tailed macaques, they are tolerant to tuberculosis. When they are infected, only 50% of them show the symptom and 10% of them show subclinical or latent period. In Thailand, it is high prevalence of tuberculosis in our country, and it is counted as top 14 highest burden countries. And Thai government set up National Ending TB Strategy in the year 2017 to 2021. Their goal within 20 years from the year 2015 to 2035 is that the number of TB deaths should be reduced for 95%, and the incidence rate should be reduced for 90%. However, the focus on this NTB strategy is only at the human's angle. Actually, long-tailed macaques can be infected with mycobacterium. That is, 75% of them are infected with mycobacterium tuberculosis that came from humans. As you can see from the right-hand side, there are many locations in Thailand that you can see long-tailed macaques living interface with humans. They are overpopulated and highly contact with humans. Thus, very high possibility for tuberculosis to be transmitted from humans to monkeys or from monkeys to humans. From our genetic data of the genetic admixture between rhesus and long-tailed macaques and susceptibility on tuberculosis, we survey for epidemiology of TB in rhesus and long-tailed macaques in Thailand. We collected specimens from 20 populations, in total 1,194 monkeys. We determined TB from 3 kinds of specimens: thrust, buccal, and fecal samples, using RT-PCR technique. As you can see here for the distribution map, rhesus macaques in green color and long-tailed macaque is in red color. From the map, you will see that all 2 populations of rhesus macaque are detected TB, but only northern population of long-tailed macaques, or 4 out of 15 populations, are infected with TB. However, as you can see in this slide, Macaca fascicularis or long-tailed macaque, they actually can be classified into 10 subspecies. And in Thailand, we have 3 subspecies of them. That is Macaca fascicularis fascicularis, Macaca fascicularis aurea, and Macaca fascicularis atriceps. The subspecies that I talked previously for the hybrid with rhesus macaque are Macaca fascicularis fascicularis. And the subspecies that I will move forward, it is Macaca fascicularis aurea. Macaca fascicularis aurea has the distribution from Myanmar to southwestern Thailand along the Andaman Sea coast. Thus, their common name is Burmese long-tailed macaques. Burmese long-tailed macaques is one of the 3 species of non-human primates from totally 505 species around the world. Just only these 3 species that they can use stone tooth to forage for foods. Another 2 species are capuchin and chimpanzee. Capuchin is a representative of American continent. Chimpanzee is a representative of Africa continent. And Burmese long-tailed macaque is a representative of Asia continent. On this slide, you will see that all three populations of Burmese long-tailed macaques are TB infected. So, and all of them are stone 2 user. So if we move to the distribution map of Macaca fascicularis aurea, and you will see that Macaca fascicularis aurea, the distribution is in light blue color, and the neighboring subspecies Macaca fascicularis is painted in light green color. And you will see that along southwestern parts of Thailand, all of Macaca fascicularis aurea or Burmese long-tailed macaque, they are stone 2 user. So by the distribution of two subspecies that they live close together, and then the hybridization between two subspecies was reported at 12 to 8 degrees north. Generally, we can identify Macaca fascicularis aurea and Macaca fascicularis by using the arrangement of the cheek hair. Macaca fascicularis has the arrangement of the cheek hair that lie across the zygomatic bone at the cheek, so-called transzygomatic. But the Macaca fascicularis aurea, the cheek hair arrangement is lie under the zygomatic bone, so-called infrazygomatic. From our study based on mitochondrial DNA, whole mitochondrial DNA, and whole genome sequence, we found that Macaca fascicularis aurea's genetics are very distinctive and different from Macaca fascicularis and Macaca mulatta. From the first one, if we look at the genetics based on partial mitochondrial DNA, you will see from the phylogenetic tree that Macaca mulatta is in green color, Macaca fascicularis is in red color, and Macaca fascicularis aurea is in blue color. So you will see that the clade of Macaca fascicularis aurea is separated from Macaca mulatta and Macaca fascicularis. So then, if we look deeper to the whole mitochondrial DNA sequence, so in general, for the people who study macaques at the moment, we have 23 species of macaques in the world, and we can classify them into 4 species groups. That is Solanus sylvanus species group, fascicularis species group, Syniga species group, and Arctoides species group. In general, Macaca mulatta and Macaca fascicularis, they all belong to fascicularis species group. But based on the whole mitochondrial DNA, you will see that for Macaca fascicularis species group, the species that you can see here is Macaca fascicularis and Macaca mulatta. But for Macaca fascicularis aurea, they fall into Syniga species group. Surprising, huh? So then we move into the whole genome sequence. It's nearly similar result. You will see that mulatta group, fascicularis group, and fascicularis aurea. So they separate into 3 cases. And aurea is separate first from mulatta and fascicularis. So it means that even though people classify them into subspecies level, but their genetics is beyond species. So now we try to raise them up to be new species of macaque. So as I mentioned in the previous slide, that the National Primate Research Center of Thailand has 525 Thai long-tailed macaques. This is because there is a high global demand for long-tailed macaques, especially during the COVID-19 pandemic. In America, pharmaceutical company is the main user for these species of laboratory animals in each year, ranging by 50 to 70,000 animals that they use, and China is the main exporter for this animal. However, long-tailed macaques are not native to China. Most of the Chinese long-tailed macaques have been illegally obtained from Laos, Cambodia, Vietnam, and Thailand. Therefore, their genetic traits are uncertain and lead to problematic results during pharmaceutical testing regarding 3R's principle for laboratory animal use. Besides, long-tailed macaques in green color in Thailand map, living across Vietnam, Thailand, Laos, and Myanmar, have a historical background of hybridization with rhesus macaque red color in the map. Then, long-tailed macaques from different locations has different genetic trait. Other than that, long-tailed macaques from different locations that they have different genetic trait, they can be classified into 10 subspecies, of which their genetics between subspecies are quite different. Then the production of macaques with known origins, clear genetic trait, and specific pathogen-free or SPF condition is very important for biomedical research, and it is the goal of our primate center. So that is all for my presentation. And I would like to thank all of my collaborators and grant agents. So you will see that on my slide there are many helps from many universities and many institutes around the world. Thank you very much for your attention.